Hello students. So today we are here with a new topic on immunology, major histocompatibility complex. This covers your syllabus of UG semester 6, DSC 4. Now this is a new topic for you and I have tried my best to make you understand the concept in a very simplistic manner. Now, since it is a topic of your immune system, you first need to understand what is basically the function of our immune system. We all know immune system works to attack an invading parasite. Now, this attacking thing is not always a parasite. It can be a microorganism, it can be any kind of self-antigen of the body, it can be a product of your metabolism, anything that can harm your immune system. Now, whenever your immune system is harmed, then the cells of our immune system will start reacting to those foreign particles so that the steady state of the physiology of our body is maintained properly. In this topic, while we are studying about the major histocompatibility complex, we need to understand primarily that the body's own tissue and foreign tissue are something towards which our immune system acts. Your immune cells or your immune system should identify whether it is acting against body's own tissue or a foreign tissue. If it is acting against my own body cell, it will be called self, and if it is acting on a foreign tissue, that will be called non-self. So I hope you understand the difference between a self or a non-self tissue. The immune system, when it identifies the body's own tissue or a foreign tissue, then this identification or recognition is partly controlled by a set of genes. These set of genes are present in major histocompatibility complex. So in a way you can say that these major histocompatibility complex are portions in the DNA or in the genes where they have such genes which can identify self or non-self tissue to fight with. Now if I need to define what is MHC, then I can say that MHC is a large region of DNA. It spans about 4 million base pairs in humans. Please mark it importantly. It spans about 4 million base pairs in humans and contains nearly 200 coding loci. This particular data is based on vertebrates. So MHC is a portion in the DNA that contains 200 coding loci. What does this mean? Like there are positions on the MHC complexes that have the capacity of recognition, and these are nearly 200 in number. Now MHC can be divided into various types, MHC 1 and 2. Now they are polymorphic, like they don't have a specific structure, they are polymorphic in nature. You have MHC 3 also, but MHC 3 is not in your curriculum, so we are not discussing MHC 3. Now what is the real function of the MHC genes? The MHC genes encode cell surface proteins that present small peptide antigens to T cells. What do? They will only encode those proteins which are on the cell surface. What do? They will present the small peptide that is in the form of the antigens to the T cells. These T cells are our immune cells that helps our body to fight infection. Hence, it is very important to understand that MHC act as antigen presenting structure. Like on a cell, if there is an infection, then on the cell, the MHC genetic system is going to provide you with certain structures that makes your T cell recognize that they are the antigens and they have to fight against them. So MSC acts as antigen presenting structures. Let us go to a brief history of MSC complex. So Gorer, a scientist in 1930s, he was working on rejection of foreign tissue. Now, what is this rejection of foreign tissue? Now, you must have heard about transplantations, the heart transplantation, kidney transplantation, liver transplantation, and several other things. Now, this is a big revolution in the field of medical science that if I have a dysfunction of a particular organ, or some diseased condition in a particular organ, then there are specific organs which can be transplanted. Now, when you have a transplantation, many a times what happens that the body rejects the foreign tissue inside the body. Now, this rejection is based on result of an immune response. You must have heard that a patient suffering with a kidney dysfunction might have a kidney transplant. In the beginning, things may seem all right, but slowly you'll see certain kind of symptoms developing in the patient, and slowly and slowly the person will again going back in the diseased condition. On investigation, you can find out that the kidney that has been transplanted is actually not functioning properly. This is because of the body is not accepting the transplanted kidney. This rejection of the tissue. So anything that is coming from outside, although you match certain things, then only we transplant, but still if it's coming from an external source, from a foreign body, that will be a foreign thing inside my body. So every time this is not happening, but many a times we can see that there is tissue rejection by our own self tissues. So Gorer was basically working on this thing. 1940s and 1950s, Gorer and Snell, they found out antigens that were encoded by the genes in the group took part in the rejection of transplanted tumors and other tissues. So he called these genes histocompatibility genes. He said that these histocompatibility genes actually were showing that something absurd was happening and the body was not accepting it. Snell was awarded Nobel Prize in 1980 for this very thing. So this is a very, very important milestone in the medical history also. So let us view certain points related to your MHC molecules. Let me remind you, MHC stands for Major Histocompatibility Complex. Major, you know, something that is in large numbers or is very important part of your thing, that is major. Histo is tissue, compatibility is how much, easily you are able to relate to certain things. So compatibility is like two individuals, if they can share things together, if they have an understanding, we say they are compatible. So these MHC, this tissue is how much the tissue that is coming from outside or your own self, how much compatible it is towards your body is the histocompatibility. And complex, why complex? Because it is a structural domain. It has got a particular structure. That is why it is called the complex. So MHC molecules are always membrane bound. They will be always seen bounded in the membrane. They are recognized by T cells of our body. Remember, they are always, maximum times, they are recognized by T cells of my body. And because they are cell surface proteins, they will always require cell-cell contact. Now, peptide from cytosol associated with class 1 MHC and is recognized by TC cells. So, TC cells stands for cytotoxic cells. Now, you know we have two classes of MHC, class 1 as well as class 2. So, class 1 MHC will be recognized, both of them will be recognized by T cells. But what is the difference? Class 1 MHC will be recognized by TC cells, that is the cytotoxic cells, and class 2 MHC will be recognized by the TH cell, which are also known as thymus cells. Now, you know, the working of MHC, the mechanism of work of MHC actually involves two levels, level 1 and level 2. First of all, what will happen? There should be a peptide molecule. This peptide molecule can be anything, antigen kind of thing. Now, this must associate with a given MHC of an individual. Now, the peptide, the antigen or the peptide should first associate with an MHC molecule. If this association is not occurring, no immune response will be seen. This is first level. So, in the beginning, what has to be done? The peptide should be associated with the MHC. Second, now when the MHC and the peptide, like I said, the antigen, when they are forming a complex, now the T cells are going to recognize this peptide associated with the MHC. So when the T cells recognize the association of the peptide with the MHC, they will start their action and will bring their sources to fight the infection. This is a second level control. And every MHC molecule will be having one binding site. This is important for your objective also. So every MHC molecule has only one binding site. This is a figure that shows presentation of MHC 1 and 2 in a cell. So you can see it's as I told you, it's a membrane bound molecule. So you can see the phospholipid bilayer of the plasma membrane. You must have studied the fluid mosaic model of the plasma membrane. So it's a phospholipid bilayer. So you can see two kinds, MHC class 1 as well as MHC class 2. Now MHC class 1 is there for all nucleated body cells, whereas MHC class 2 is there for the macrophages, the dendritic cells and the B cells. You know macrophages, they are very, very, active cells in killing infections. They have again, these are polymorphic, so they can change their shape, they can catch the infection and they just engulf them. You know the dendritic cells that comes from the nerve cells and the B cells of your body that are again involved in maintenance of our immune system. So if you can see the structure, the MHC class 1 has alpha 1, alpha 2, alpha 3 and 1 beta microglobulin, whereas MHC class 2 has 2 alpha 1 and alpha 2 units and 2 beta 1 and beta 2 units. You can see the antigen binding cleft in both MHC class 1 and class 2. So this is the position or the groove where the antigen will be binding up with the MHC class 1 or class 2 molecule depending on the kind of tissue. Class 1 MHC genes encode glycoprotein. I hope all of you understand a glycoprotein. So there are proteins on your plasma membranes, and when they have the carbohydrate molecule on them, they are known as the glycoprotein molecules. So they encode the glycoprotein expressed on the surface of nearly all nucleated cells. Whatever nucleated cells you have in your body, the glycoprotein on them is encoded by the class 1 MHC gene. So what do they do? What is the product? It is the presentation of peptide antigens to TC cells. What is this presentation of peptide antigens? I told you there will be an antigen that is coming from body's own self or foreign tissue. This antigen or the peptide molecule is going to bind with the MHC. Now this association of the MHC as well as the peptide will now be identified by the T cells. TC cells, I mean the cytotoxic cells. On the other hand, in class 2 MHC, they will be the TH cell. So mechanism is same for both of them. Class 1 MHC will always include nucleated cells. Class 2 MHC is going to include the dendritic cells, B cells and the macrophages. Class 2 MHC will be having the thymus cells for recognition, and class 1 MHC will be having the cytotoxic cells for recognition. Now class 1 MHC, the presentation of foreign intracellular antigens. Foreign intracellular means intracellular, that is within my own cell, but foreign, my body is not able to identify it. Or the altered self antigens. There are certain antigens in my body that are produced by my own body. So they are called self antigens. These are the targets for destruction. Whereas in class 2 MHC, these are extracellular foreign antigens that are coming from outside the body. So whenever a foreign particle will be coming from outside the body, what will be the immune response? It will be production of antibodies. But if it is from my own body, inside my body, through the foreign intracellular antigens, then the cells are destroyed. There is no antibody production. So remember, antibody production will only take place in class 2 MHC proteins, not in class 1 MHC. Why? Because in class 2 MHC, you have a foreign particle that is entering your body. Whereas in MHC1 system, most of the time, the foreign thing is an intracellular substance. This is a small differentiation between the class 1 MHC and class 2 MHC. Whatever we have studied, just a simple recapitulation of that. So polypeptide chains in class 1, they are alpha and beta 2. Okay, so one beta 2 microglobulin and then you have 3 units of alpha. Whereas in class 2 MHC, you have 2 alpha units and 2 beta units. In class 1 MHC, you have alpha 1 and alpha 2 domains. Class 2 MHC, alpha 1 and beta 1 domains, as you had seen in the figure. In the binding site in class 1 MHC is CD8. CD8, again CD8 are those cells which are the cytotoxic cells. And the CD4 will be in class 2 MHC. We'll be discussing this furthermore. Now, size of peptide binding cleft. What is the peptide binding cleft? I had shown you the antigen groove. That is what is the peptide binding cleft. So, in class 1 MHC, it will nearly accommodate 8 to 11 residues. Whereas in class 2 MHC, it will include 10 to 30 residues or even more than that. Now, class 1, you have some certain nomenclature for class 1 MHC. In human, we call it HLA, that is human leukocyte antigens. So it's HLA-AA, HLA-B and HLA-C. And in class 2 MHC, it's HLA-DR, HLA-DQ, HLA-DP. You need to know the names. And in mouse, it's H2, H2D, H2L in class 1 MHC. And for class 2 MHC, IA and IA. Now we are studying in mouse also because graft rejection, tissue rejection, a lot of investigative studies are done on mouse, on a mammalian tissue. So actually the many medical findings are based on the investigation of mouse, and so we have a data on mouse. So this is a general idea of what are MHC molecules, why are they set class 1 and class 2, what is the difference between class 1 and class 2, and a simple outline of how they work. So I'll be dealing more with this in my second presentation that will be continued. Like I'll be discussing the exogenous and endogenous pathway of these systems. So I hope this will be of great help to you. Thank you so much, students. Stay safe, stay home. Thank you.